So welcome to the class. We will review cell cycle and cell division in this lecture. Perhaps it's important that we start with the definition of cell cycle. We define cell cycle as self-regulated sequence of events that occur in a cell. And this self-regulated sequence of events are aimed at two things. They are aimed at controlling the growth of that particular cell, but they're also aimed at leading that cell to its division so that the cell eventually divides into two daughter cells. So that self-regulated sequence of events is what we are calling the cell cycle. It is a continuous process if you get it right, because a cell will be doing this, then the cell divides, and the daughter cells begin the journey again. We routinely describe the cell cycle as having two major phases. One of the phases of the cell cycle is known as the interphase. If you understood that it is a sequence of events that lead to its growth and its division, then we can define the interphase as the period between two successive cell divisions. The period between two successive cell divisions. During that period, the cell largely grows and also replicates its genetic material. And then the other phase is what we call the M phase. The letter M standing for mitotic phase, which is now the period of actual division. And during mitosis, the cell separates. There is division of the nucleus, as well as division of the cytoplasmic contents. So the nucleus will divide, aimed at going to two daughter cells, and then the cytoplasm also develops the nuclei that are going apart, and that is what happens during the mitotic phase. So the cell is either dividing or is in that period of preparing to divide. So interphase is that period of preparation for division. Then M phase is the actual period of division. Perhaps let's say something more about the interphase. We have already given it the definition, the period between two successive cell divisions. So if you look at this image, if this is the period of cell division, this period between one division and the next division is what we're calling the interphase. During that time, immediately a cell has been formed from cell division, the cell begins to grow. And it is during that time that the cell will also replicate its genetic material. Perhaps you're asking why the cell needs to replicate its genetic material. Let's think for a moment that it is possible to count the DNA that a cell has. And then let's also assume that we are able to count and we found that each cell has 200. Or let's say the cell that we have in question, the cell that needs to divide has 200 DNAs, and I'm taking this literally so that we are able to understand what I want to put across. If this cell has 200 DNAs and this cell divides, would expect the daughter cells then to have 100 each. Now, do you imagine what will happen when the daughter cells then divide? The granddaughter cells will have 50 each. Now think through that over several generations, then you'll realize that cells will actually be losing their genetic content. Now, we don't want that to happen. Otherwise, will not be in existence in our generation. So a cell that is intending to divide, this cell we are starting with that has 200 DNAs that is intending to divide, for it to make sure that the daughter cells will also have the same amount of genetic material that it has, the cell that is intending to divide must replicate its genetic material. So in the example I've given, if this cell has 200 DNAs, it will replicate that so that it has now 400 DNAs. To replicate is just to make another copy of itself. The DNA is synthesized so that it makes another copy of itself. So that instead of 200, now we work with 400. And inevitably then, this cell divides. When it divides, the daughter cells will each have 200, just like the original genetic makeup of the parent cell. Now, this is the rule of genetics. This is the rule that a cell that needs to divide has to replicate its DNA before that division. This is to aim at ensuring that the daughter cells have the same amount of genetic material as the parent cell. So during interphase, that's when the cell will actually do that. It's part of preparation for cell division. Routinely, we divide interphase into 3 stages. There's what we call the G1 phase, S phase, and the G2 phase. Let's start with the G1 phase. The G1 phase of the cell cycle, that letter G stands for gap. So we call the gap 1 phase. So this is the period immediately after cell division. The period immediately after a cell has been formed, a new cell. Immediately a cell has been formed, the cell will begin to grow. Because remember, it could be slightly tinier than what we intend it to be, because cytoplasmic division has taken place. So the cell will grow and perhaps accumulate nutrients. But remember, this cell is intending to divide later, some days. So because it is intending, it's looking forward to division, it means that at some point it will have to replicate its genetic material. And if this cell is going to replicate its genetic material, then it needs the correct machinery that will regulate the process of DNA replication. So this is the time that the cell prepares the necessary machinery that is required to control DNA replication. There's some molecules which are formed in this stage, which are vital in checking a number of things in the cell cycle to ensure that things are okay. Perhaps it's important for you to also know that if a cell successfully goes through the G1 phase, most likely that cell will divide. And so if the process of the G1 phase is not correctly checked, there could be some problems that can be seen as with regard to, for example, the development of cancer can be just because of some defects which occur in the G1 phase of the cell cycle. Okay, let's assume normal, that the cell has gone beyond the G1 phase successfully. It now has the necessary regulatory molecules that will help it to go through DNA replication. Then the cell will then take up that particular activity, DNA replication, and the period of DNA replication is what we call the S phase. The letter S in this case stands for synthesis. This is the synthetic phase. And what are we synthesizing? The DNA is what is being synthesized. So the S phase is a period of DNA replication. At this time, the genetic material is doubled for the reason that I've just given you a few minutes ago. In that case, therefore, it means that when we are going to have chromosomes, a chromosome will be having 2 chromatids, because now we've made an extra set of the genetic material. So each chromosome at this stage will be, later on, as we're going to see, the chromosomes will be having 2 chromatids each. Great. There are some checkpoints again in the S phase that just confirm that the DNA that has been replicated is normal. And in case there's need, then some repairs actually take place. And perhaps even cell death can take place if those repairs are very grievous. Once a cell has replicated its DNA, remember it's aimed at going to divide eventually. This cell will then go to the next phase. Well, the image here shows you the process of DNA replication. So the original DNA usually first must unwind itself. Once it has unwound itself, then each strand that has been formed then forms another complementary strand. With that in mind, therefore, usually the DNA that is being formed, the new DNA, will actually be an exact copy of the original genetic material. So in DNA replication, we don't change things. The DNA that we have is exactly similar with the original. Okay. After the S phase, the cell goes into the G2 phase. As I told you, G stands for gap. So the gap2 phase. The gap2 phase of the cell cycle, this part here, is a period after DNA replication. It is also a period when the cell will continue to grow. And importantly also, it is also a period when organelles will also be formed and perhaps be reorganized because we are preparing for cell division. One of those organelles which will be reorganized will include the centrioles, really. Remember, they are formed from microtubules, which are important in cell division. So basically, gap2 phase is that period when it's actually preparing for division. Well, there's also some check that occur during the gap2 phase of the cell cycle. Basically checking whether the DNA that the cell has is normal and things like that. After the G2 phase, that means that interphase is over. Now the cell enters the mitotic phase. We call it the M phase, M for mitosis. The mitotic phase is a period when the nucleus divides, and after the nucleus divided, the content of the cytoplasm of the cell also divide. So it's a period of nuclear division as well as cytoplasmic division. We routinely divide mitosis into stages. There's the prophase, metaphase, anaphase, and telophase stages of mitosis. Perhaps a reminder of what happened in those phases will be good. So I'll not go into deep because you've maybe you already know this from high school. The key thing that occurs during prophase is that the genetic material condenses into visible chromosomes. So we now have chromosomes. The genetic material is condensed into chromosomes. Because the genetic material was replicated, we expect, therefore, that we have 2 copies of the genetic material in each chromosome. And those 2 copies are the ones we are calling the chromatids. Apart from the condensation of the genetic material to form chromosomes, we also have the disintegration of the nuclear envelope. So the nuclear envelope disintegrates during this period. Well, there are other things that may also happen as concerning the microtubules which form the centrioles. They may be moving apart in preparation for this particular process. The hallmark of the prophase is that the genetic material condenses into visible chromosomes. After prophase, the next stage is metaphase. This image displays the hallmark of metaphase. During metaphase, those chromosomes which have already been formed or are now visible during prophase now realign themselves in the equator of the cell. So the chromosomes align themselves in the equator of the cell. That's the hallmark. At this point, you note that the centrioles have now gone away on extreme end because they are preparing now to split this particular chromosomes. So that time when the chromosomes align in the equator, perhaps could also be the best time that karyotyping can be done. Remember, karyotyping is a graphic representation of the chromosomes within the nucleus of a cell. So this is the best time that karyotyping is done. You are able to study the chromosomes at this time. After metaphase, the next step is anaphase. Anaphase is that period when the chromatids are separated. As we can see in this third image, the chromatids are being separated. So one chromatid is pulled to the other pole, while the other chromatid is pulled to the other pole. Remember, the chromatids were of a single chromosome, but this is because the genetic material was already duplicated. So now they're being separated. Sister chromatids are being separated at this time. Once the sister chromatids have been separated, well, now we have telophase. During telophase, now the cytoplasm is the one that is now trying to separate. As you can see here, now the cytoplasm is trying to separate. Also, the nuclear envelope is now reconstituting again. That's telophase. So those are the stages of mitosis. Of course, eventually the 2 cells are going to go apart in what we call, well, completing the process of cell division. So why do cells undergo mitotic cell division? Cells undergo mitotic cell division. One, because we desire to achieve some numerical increase in the number of cells. Think about blood cells in your bone marrow. We just want numbers and numbers, and faster for that matter. So when you want numbers and faster for that matter, mitosis is a good way of increasing the number of cells. We just want it for proliferation. Cells also undergo mitosis because through this, the organism or an organ will actually grow and develop. Think about the size you were when you're born, and right now, you've increased in size, you've developed thanks to mitosis. So mitotic division is a mechanism of growth and development, either of an organ or an organism as a whole. Importantly also, mitosis is a mechanism of repairing worn out tissues or worn out parts. It's a mechanism of replacing what is gone through wear and tear. Think of a wound that could be there. Later on, there is some healing. Mitosis is important for this. Importantly, note that in mitosis, the original genetic makeup is maintained. So the daughter cells have the same amount of genetic material as the parent cell. The daughter cells will have 46 chromosomes, just like the parent cell having 46 chromosomes. Great. How about if it was meiosis? Meiotic cell division is unique. It only occurs in the germ cells. So it will affect the sperm and the oocyte only. Meiotic cell division involves two phases as well. There is meiosis 1 and meiosis 2. In the first phase of meiosis, the main thing is separation of homologous chromosomes. We know that we have 46 chromosomes. Put in other words, we have 23 pairs of chromosomes. During meiosis 1, the homologous chromosomes are the ones which are separated, which means that we separate the pairs of chromosomes so that each daughter cell after meiosis 1 has only 23 chromosomes. That's what we call haploidy. So the aim of meiosis 1 is to achieve haploidy. Then meiosis 2 is very similar to mitosis. The only difference is that in meiosis 2, we are working with 23 chromosomes, unlike mitosis, where you're working with 46 chromosomes. Now, you need to ask yourself why cells need to divide meiotically. Meiotic cell division serves two key roles. One, meiotic cell division achieves haploidy. The daughter cells are haploid cells. They have 23 chromosomes instead of 46 chromosomes. They have only one set of chromosomes instead of two sets of chromosomes that we usually see in the diploid cells. And perhaps you're asking yourself, so why is it important to then have haploidy? Remember, these are the germ cells, the cells that will eventually meet at fertilization, so that now we then form a zygote and yet, yes, another human being formed. The reason why we make these cells to have haploidy, to be in the haploid state, is so that when fertilization takes place, we can restore the normal diploid number of chromosomes. So we need to have their genetic material so that at fertilization, we restore to normal. Otherwise, if we didn't do this, then it means that every time human beings get offsprings, the offsprings will be having more genetic material than the parent cell. So again, it's a mechanism of ensuring that we have the same number of chromosomes throughout generations. Now, the second reason why cells undergo meiotic cell division is so that we can have genetic variation. The daughter cells in meiosis are not genetically identical, unlike in mitosis, where the two daughter cells are actually genetically identical. In meiosis, the four daughter cells are not genetically identical because there's some crossing over of genetic material during meiosis. And because of that, we'll have genetic variations. So that is meiotic cell division. You need to distinguish it from mitotic cell division. Now, how do you account for those cells which have lost their capacity for cell division? Yes, we've talked about the cell cycle and we've said that there is mitotic phase and interphase, and that interphase has gap 1, S phase, and gap 2 phase. And then mitosis again. How about those cells which no longer have the capacity for cell division? The cells which have lost their capacity for cell division are described as being in the G0 phase of the cycle. Now, the 0 here actually means O, not 0, and that O means outside the cycle. We believe that these cells have now left the cycle. So G0 phase is a phase of terminal differentiation. The cells have lost the capacity for cell division. They can no longer divide again. Well, in some circumstances, the cells which are in the G0 phase may be reintroduced into the cycle. But that induction, so that they can be reintroduced into the cycle, is in most cases an abnormal induction, like in the case of cancer cells, in most cases. In a normal scenario, they will have lost the capacity for cell division, and so they rest. We call the quiescent stage. Some examples of cells which are in this phase include mature red blood cells. You know, they don't even have a nucleus, so they can't even divide. Skeletal muscle cells, they don't divide. Most nerves, they don't divide. A number of cardiac muscle cells, if not all of them, they don't divide. So they are considered to be in the G0 phase. The mature cells of bone, we call them osteocytes, they don't divide. Great, so that is cell cycle and cell division. That's what I had for you in that particular lecture.